Hello, my name is Kim Bertay. Today we'll be getting into the Introduction of Microbiology lecture series. We want to go ahead and start looking at what this class entails. We could look at a couple different topics. So we can start out with microbial pathogens, which is typically the first thing we think about when we get into microbiology. But what we'll see from this lecture series is that pathogens are not the only thing that we'll be focusing on. Actually, a large part of our microbiome, the microbes that live on us and within us, are actually beneficial to our survival. We'll also be looking at the role of microbes in the development of our immune system. We'll be covering the immune system from adaptive to innate. We'll be looking at different disease symptoms and signs as well. Okay, so looking at the first section, we're going to see how microbes have shaped history throughout time. Microbes were first related to human health, so we saw implications in human health. We're going to see how cautious postulates were able to actually bring together microorganisms and their impact on humans. And we'll also get into microbes within the environment and how that's important for human life. Okay, let's go ahead and talk about where microbes came from. So microbes are the simplest form of life. They were the first to evolve. This image that you see on the left hand of the screen is actually a mural, a picture taken from the La Brea tar pits in Los Angeles, California. And what we see is an environment billions of years ago on Earth. And what we see is that Earth is not the way we see it today. So here we see lava, we see thunder, thunder and lightning storms. And we see that in this harsh environment, in this extreme environment, that molecules start forming and coming together. Some of these molecules that you can see here, you might notice as macromolecules such as DNA or carbohydrates or lipids or even proteins. These are the essential molecules that we use that form cells. So these elementary molecules build up together, create cells, and cells start interacting with one another, and we start getting multicellular life as well. So communication between cells and within cells for multicellular life. As we move down to the Cambrian explosion, we can see multicellular life forming in the water. Then later, multicellular life coming to land and ending up where we are today. So really, the story of microbiology, the evolution of microorganisms, is part of our story as well, that we actually come from these precursor reactions that we found on Earth. So cells, again, are the smallest form of life, and that's what we're composed of. Cells, we're just multicellular organisms. Okay. When we look at microbes, we can start asking ourselves, well, what do they do? In the environment, they can be extremely important because they can fix nitrogen. So nitrogen is not always readily available for plants to use, and it's essential for plant life, which we're dependent on as well. So they can fix nitrogen from the soil and make this nitrogen readily available for plants. Microbes, even within our gastrointestinal tract, can produce vitamins, like E. coli can make vitamin K within our gastrointestinal tract. And microbes themselves, like the one you see on the right, can be photosynthetic. So we see this as a green microorganism, and they can produce food for the rest of the food chain through the process of photosynthesis, harvesting light energy from the sun and converting it into glucose, simple food. So not only do they support the food web and create nutrients for ourselves, we also think of microbes from the pathogenic standpoint of the ones that cause disease. And of course, in microbiology, these were some of the first organisms to be studied, but we don't want to forget the positive benefits of microorganisms as well. When we talk about a microbe itself, a microbe is going to be defined as something that we need a microscope to see. And that's actually a wide range of organisms. So even we're looking at helminths or we're looking at other microorganisms, sometimes it's actually their offspring or their eggs that are microscopic. And so we categorize them in the field of microbiology. The cell itself, which makes up microorganisms, a single cell, is the smallest form of life. And we're going to see all the metabolic reactions that are enclosed within a membrane. So we'll see all of these chemical reactions occurring, which make life possible. Beyond a cell, we have viruses, and we'll discuss viruses a little bit more as non-living components of microbiology. Looking at different kinds of microbes, we have many, many different species. They're extremely diverse. They're actually the most diverse living organisms that we know of. When we talk about microorganisms, we have to be able to organize them. So we can organize them by genus and species. So here we see the term Staphylococcus aureus. Staphylococcus is going to be the genus, and aureus is the species. When we're writing these names, we can take note. This is italicized, and Staphylococcus is uppercase for genus, while the species is lowercase, aureus. Okay. We might have heard of Staphylococcus aureus before. Maybe we've heard of MRSA or MRSA, as some call it. That's methicillin-resistant Staphylococcus aureus. Yeah, a real threat to the healthcare environment. Looking at microorganisms themselves, what can make them differentiated from one another is going to be their DNA. So just like us, their genomes are going to be unique to them, and the blueprint for life of a single cell or blueprint for life of multicellular organisms, just like us. Okay. When we compare microorganisms to one another, we can talk about prokaryotic and eukaryotic microorganisms. Prokaryotic microorganisms are what we typically think of like bacteria, but there's also archaea within that category. Prokaryotic organisms are very simple-celled organisms, and these unicellular organisms don't have membrane-bound organelles, and they don't have a nucleus. So actually, prokaryote, the term, means before the nut, so before the nucleus, the center that houses the DNA. On the other hand, eukaryotic is the microorganisms that do have a nucleus to house their DNA and membrane-bound organelles. This structure makes them quite a bit larger than prokaryotic organisms. And this category contains fungus, protozoa, and algae. What we see at the bottom left of the screen is actually an amoeba, so that is going to be a eukaryotic microorganism. Okay. Different kinds of microbes also include viruses. So these are non-cellular. These are non-cellular in the category of microbiology. We can also call them abiotic as they're not living. They're not living for quite a few reasons. One that we see here is that they're non-metabolic. Non-metabolic means they're not running metabolic reactions. So they don't have energy that they're producing. They don't have energy that they're consuming. All cells that are living are producing energy and using energy. We have catabolic reactions and anabolic reactions. We're building and breaking things down constantly. Viruses, on the other hand, don't do this, and they're quite a bit smaller because they don't have to maintain themselves and produce energy and break down energy as well. Another thing that sets them apart from what we call living organisms is the fact that they can't reproduce on their own. So they're dependent on host cell machinery, something like the DNA polymerase. So while viruses can have genetic information like DNA or RNA, they actually rely on the host cell to reproduce and to make more of their DNA. So they're non-living for that reason as well. So we don't call them alive. Now, the definition of alive is going to be changing in the future and has also changed in the past a bit too. The more and more we can classify living from non-living, the more and more exceptions we find in the sciences between viruses and unicellular organisms that we think is the smallest form of life. Okay, let's go back in time and talk about microbiology and its impact on the human population. When we think of microbiology, again, we think of the pathogens that have affected us throughout history. So we know things like tuberculosis and leprosy had a profound effect on humanity. These microscopic organisms were the ones causing disease, but it wasn't yet known at this point that microorganisms were really playing a role in disease. So others had thought Maybe this was due to behavior, this was due to bad spirits, it was due to an imbalance in the bodily fluids that we had that caused disease, or some thought it was from breathing in, inhaling impure odors, is what actually had caused disease. So microbes were not necessarily known as the infectious agents. So people were observed with the signs and symptoms of sickness, but there was no correlation to microorganisms before 1850s. Okay, when we start understanding microorganisms, it starts with the invention of the microscope. So here we see Robert Hooke was one of the first people to build a microscope. And from there, we have Anton van Leeuwenhoek, who is able to make observations of microorganisms. And the microscope he used is the one envisioned here in this image. So it's actually a microscope that's held up to the eye with a very, very small lens. Um, Anthony van Leeuwenhoek was actually not a microbiologist developing a microscope to understand microbes themselves, as it wasn't a known field at the time. He was actually a merchant, and what he dealt with was fabrics and what the thread count was on fabrics. So, in order to see the thread count on a fabric, he developed this microscope. But he also started using this microscope to look at the world around him, and his curiosity led him to discover this world of microbes. We can see some of the first images that he drew here, with Anthony van Leeuwenhoek was drawing. And what he noticed were things moving around that you could only see through a microscope that you couldn't see with the naked eye or the unaided eye. Anthony van Leeuwenhoek actually first described these living things as animalcules because it's a cross between animals and molecules. So he knew there was smaller building blocks to what we were looking at here. When microorganisms were first discovered, they were not connected to disease at the time. There was no connection between the two. Nobody saw them as relevant. It took about 200 years before we found the connection to disease. Okay, let's look at spontaneous generation. Spontaneous generation was a concept that life poofed into existence. So this was debated heavily in the 18th century, and Louis Pasteur was one to set up experiments to counteract the concept of spontaneous generation, that life just poofed into existence. And what he discovered was that life comes from pre-existing life. Louis Pasteur's experiment that you see here was set up in this S-shaped flask, curved flask here that we have. And he had a growth medium in which he grew microorganisms. So this had all of the proper nutrients available for microorganisms to grow in. And with this S shape here, it was open to air, but would not be able to contaminate the solution due to this S curve here, would trap the dust and the microbes. So what he did is he sterilized this sample with this growth medium and then looked to see if life could spontaneously occur. What he found is that life did not just spontaneously occur within the medium. So what he found is that life only comes from pre-existing life. So if microorganisms were in here before, it would lead to the generation of more microorganisms. But if he sterilized it, no microorganisms would appear out of nowhere. Spontaneous generation was actually thought of as the way life came into existence because what people saw is when food was left out for an extended period of time, that all of a sudden maggots, the larvae of flies, would just appear out of nowhere. Now we know today the larvae of flies does not appear out of nowhere. They're laid down by the fly, and these eggs hatch open. When it comes to fly larvae, what we're seeing though is that they were microscopic. So because we couldn't see them, we just had assumed that they appeared out of nowhere. We weren't able to see the microscopic reality of the situation. So Louis Pasteur's experiment disproved spontaneous generation and proved that life comes from pre-existing life. Let's go ahead and look at the germ theory of disease. So here we're going to start associating with microorganisms with disease. We know that disease often happens in overcrowded areas. So this can be in big cities. We see the spread of disease rapidly. And it was also happening during warfare times. What we see in this image here, this is actually Florence Nightingale, a nurse who worked in war times and was analyzing soldiers and what they were actually passing away of, what they succumbed to. And what she found out actually, the mortality rates of these soldiers was more likely due to infectious disease than it was to their actual battle wounds it's inflicted. So here we can see in this diagram, it says April 1855 to March 1856. In pink, we see the battle wounds that people are succumbing to, and in blue, we see infectious disease. So while the battle wound may have been the portal of entry for the microorganism, what actually led to the fatality was the infectious disease. So Florence Nightingale was one of the first people to make this association that soldiers were being lost on the battlefield, not from their battle wounds, but from infectious disease. So preventing infectious disease is how they were going to be able to save the soldiers and send them back out and keep the army numbers high. Florence Nightingale, as a woman, had a hard time with her concepts, presenting them at the time, but people actually ended up listening to her because they realized it was saving soldiers' lives. So she made a really large impact in linking infectious disease to mortality rates. Today, we have the field of public health, which understands this relationship of disease to our population. And the people studying it are called epidemiologists, and they're understanding and tracking disease, and they're implementing the concepts even from Florence Nightingale. Following that, we have the Center of Disease Control, the CDC. This is where the epidemiologists in the US work. We have the WHO, the World Health Organization as well, that has epidemiologists that help track disease worldwide. Looking at infectious disease and mortality rates, we can also talk about seasonal influenza, as the flu can kill people who are vulnerable, so people who are immunocompromised, young, or elderly. And we know that the CDC doesn't only track the flu, but it actually predicts which viral strains are going to be coming up next. So here we can look at seasonal flu and predictions of strains of organisms and which ones we should make vaccinations for. So every year we know that we have a seasonal influenza vaccine, and those are created from these predictions from the CDC. And they're linking this to the spread of disease that they're observing overall globally, and what they predict will be the biggest seasonal flus to hit. Okay, let's talk about growth of microorganisms. So how do we grow microorganisms? We can grow them in a laboratory using culture. This culture holds the necessary nutrients for microorganisms to grow. And we can also start understanding how microorganisms work and how they cause infection by being able to grow them in a pure culture. A pure culture is different than what we find in nature. In nature, we find microbes in what we call biofilms, accumulations of hundreds of different organisms together. And we can isolate just some of them and grow them. So if you wanted a specific strain of E. coli, you would need to separate it from the environment that it was in and grow it in a pure culture. And with that pure culture, you could start testing and understanding the morphology of it, the biochemical activity of it, and understand more about that specific species. Pure cultures themselves, as seen on this plate, this is a nutrient-rich medium, and what we see in red here is the microorganism. These individual dots that we see are actually called colonies. That's where one individual microorganism landed on the plate and then divided over and over and over again until it became visible to the unaided eye. So all of these microorganisms in this colony should be genetically identical for the most part. Growing pure culture is going to help out Koch link disease to infection. When we talk about Koch himself, the way that he linked disease to infection actually won him the Nobel Prize in 1905. And we're going to see how he went ahead and did this. So we have four steps in Koch's postulates here. Number one is the microbe needs to be found in all cases of the of individuals that are affected by the disease, and anyone not affected by the disease has to be clean of that microorganism, that we cannot isolate that microorganism from someone. So we have two study groups, one infected, one non-infected. The infected one has to have the signs and symptoms and have the microorganism, while the uninfected should have no signs of the microorganism. From the ones that are infected, we isolate that microorganism and we grow it on pure culture. So we're not growing the entire microbiome of this rabbit. We're only growing the microorganisms that can cause disease. From here, we take those microorganisms and we can inject them into a host. What we're trying to do here is replicate what we initially saw. Does this animal have the same signs and symptoms of the counterpart in which the microorganism was initially isolated in? If so, if it succumbs to these signs and symptoms, as we see before, or displays the same ones, can we isolate this organism of interest again? And is it the same organism that we isolated here? So this shows us that this microorganism is the one that causes this disease by repeating this experiment into a healthy individual. When it comes to Koch's postulates, these four steps work great to identify a microorganism and link it to disease in many cases, but it's been given up more in recent times because it doesn't apply to all cases. For example, if we're looking at tuberculosis, if we're looking for people infected that have signs and symptoms, well, only about 10% of the people actually infected display signs and symptoms. So it wouldn't be a good example for Koch's postulates. On the other hand, we have issues where we wouldn't be able to directly test on a model organism. So for HIV, we don't really have a model organism to test HIV on that has the same signs and symptoms, and we couldn't be testing this on humans. So we see that it can fail in a few situations. We also have more molecular techniques today that help us understand the causality between a microorganism and disease. Okay, let's back up a little bit to 100 BCE. In 100 BCE, back in India and China, people were starting to understand immunization. So they were starting to understand the exposure to microorganisms and how this could prevent future deaths. This is the initial foundation of actually vaccinations that we're looking at here. So in India and China, what people were doing was exposing a person to the pox pustulates. So pox pustulates are the actual physical manifestation of smallpox. So they were taking a portion of the pox pustulates fluid and introducing it to another person. By exposing another person to this fluid, the person's immune system was recognizing this microorganism and being able to adapt to it. In the 18th century, Lady Mary was able to bring this technique to was able to bring this practice to England and display the same kind of immunization with other people. So this again is the foundation of what we now know today as vaccinations. Further work on immunization was done by Dr. Edward Jenner, in which he didn't actually use the postulates of smallpox. What he found out is that he could use postulates of something called cowpox. And this is actually a quite interesting story, the background of how cowpox and smallpox are related. So smallpox are in humans, cowpox are in cows. So what people had observed at the time is that milkmaids, women who were milking cows, were getting small infections with cowpox, which was found on the udders of the cows that they were interacting with, but it had very minimal signs and symptoms. But the benefit of getting cowpox from a cow is that it actually prevented these people from getting devastating signs and symptoms from smallpox. So if they caught smallpox after their exposure to cowpox, their immune system was familiar with cowpox, which was similar enough to smallpox for their immune system to regulate on the signs and symptoms that would have typically been displayed with the smallpox infection. So what Edward Jenner did is create an immunization pathway by isolating fluids from cowpox and inoculating humans with them, which would then protect them from smallpox. So So this was called a vaccination. And VACA stands for cow, and that's where we get the term vaccination from these cowpox, from this observation. Okay. Louis Pasteur, not only did he disprove spontaneous generation, he also worked in vaccinations. And what he actually did was a different methodology. He took a microorganism and attenuated it. He weakened it. So by weakening them, you can grow them in a lab and weaken them where you're removing certain food from their nutrients and they start becoming weaker, not as effective as microorganisms. So by attenuating them or weakening them, you could inject these weakened microorganisms into people, and the person's immune system would be able to capture the microorganisms and create immunity from there. Okay. Looking at how we've adapted to microbiology over the years, we can look in the past at what was going on in operating rooms. If you could imagine being in an operating room where nobody had a mask on, nobody had gloves, there was no proper PPE, that's what originally operating rooms were composed of. Today, we have a very, very different view, and this is in part due to Ignaz Semmelweis, who discovered really how to use antiseptics in hospitals. So what he noticed was that in hospitals where women were delivering babies, that the women who were delivering babies were more likely to die of an infectious disease following birth than women who were giving birth at home with midwives. So this was really interesting because there's a socioeconomic difference, and they found that people with more money who were giving birth within the hospitals were actually more likely to succumb to illness and die than the women who were giving birth at home. So when he followed what doctors were actually doing and why these women were more susceptible to infection, he found it was because it was the lack of hygiene within the hospitals. So the doctors who were delivering babies were actually the same doctors who were performing autopsies. At this time, there was no such thing as gloves, so autopsies were done just by hand. And people were not washing their hands following autopsies where somebody maybe died of a bacterial infection. And then they were moving into the maternity ward where they were helping women and assisting women in giving birth. And they were infecting these women who were giving birth. This doctor actually recommended, he was very young at the time, but recommended that doctors wash their hands. And he was really ridiculed until the end of his life. And antiseptics were not something that were initiated in hospitals during his lifetime. But it did lead to, his initial understanding of this did lead to large changes after his passing. We have Joseph Lister as well, who started using chemical treatments on surgical instruments. If you can imagine, surgical instruments were just used from one patient to the next. Same thing with blood transfusions. They were using the same needles on people. So this chemical treatment of surgical equipment and things used within the hospital started to come about, which prevented the spread of disease from patient to patient. And now today we have aseptic operating rooms because of these studies. Let's go ahead and look at antibiotics. So interesting story about antibiotics. You may be familiar with penicillin. Well, penicillin was accidentally discovered by Alexander Fleming. It was also accidentally discovered prior to Alexander Fleming before. What he was actually doing was working with microorganisms in a laboratory. Here we can see he has petri dishes, this nutrient-rich media, which is used to culture microorganisms, to grow microorganisms. So he was growing microorganisms, as we see these lines and dots here, on a petri dish, a nutrient-rich medium. But he sees that he has some contamination on his plate. So what we see here is kind of a fuzzy mold growth. This is a penicillium mold. It was growing on his plates. So while he was running experiments, it seems like this ruined his experiments, but actually in reality, what he ended up discovering is that this penicillium mold wasn't allowing bacteria to grow around it. This is what we call a zone of inhibition. It was inhibiting bacterial growth. So the bacterial frontier slowed as it came close to the penicillin. So what he realized is this penicillium, which is a mold, it's a fungus, was actually preventing the growth of bacteria. And that was the first antibiotic discovered. Here we have Florey and Chain were able to purify the penicillium that was this penicillium fungus, and then able to actually extract penicillin from it, which was causing the microbes not to grow. And that was used in treatment. Interesting story. The first person to be treated with penicillium was penicillin was a police officer that cut himself on a rose bush outside of his house. That cut became infected, and penicillin was grown in large scale, and they were purifying the penicillin antibiotic and administering it to this patient. Now, unfortunately, they didn't have enough penicillin. Um, Successfully isolated and purified to give them. So they actually had to take his urine, purify it from his urine, and issue it again. Now, unfortunately, the first patient did not make it and succumb to the bacterial infection, but it was a good example of the use of an antibiotic. And later, we learned how to mass produce penicillin. And this really gave birth to the pharmaceutical industry. Let's look a little bit into viruses. So we said viruses are non-living. Viruses are interesting because they're similar to our single-celled microorganisms. They're made out of the same thing. So they have proteins, they have lipids, they have carbohydrates, and they have nucleic acid like DNA or RNA within them. But they're non-living for multiple reasons that we previously discussed. When people were starting to understand bacteria, is when we started to also understand viruses as well. So these were known as infective particles. So we're looking at pathogenesis itself. One of the first viruses ever isolated and identified was the tobacco mosaic virus. So this is a virus that affects tobacco plants. So viruses don't only affect animals, they also affect plants, and we'll see later that they do affect even bacteria and archaea. So viruses affect all forms of life. As you can imagine, the tobacco mosaic virus itself was quite interesting to study because it was killing all of the tobacco plants. So there was quite a lot of money poured in to understanding how to keep these crops healthy at the time. Okay. When we look at microorganisms in natural ecosystems, they are plentiful. They are everywhere. Whether we're looking at saltwater environments, whether we're looking at brackish water, whether we're looking at pure water and lakes, we find microorganisms. Whether we're looking at terrestrial environments, or even whether we're looking at even looking at the atmosphere, we find microorganisms. They are ubiquitous as they are in all of these environments. So scientists started to study these environments and the benefits of microorganisms within these unique ecosystems. So within these niches, we see that microorganisms were able to work with one another. So some of them were working with organic molecules, some of them were working with inorganic molecules, and they all seem to fit together and play a role in this unique ecosystem, whether it be in an aquatic environment, terrestrial environment, and what we're learning about today more, the atmosphere, which is actually what I study in microbiology, aeromicrobiology, and the role of microorganisms in this niche environment. In order to take microorganisms out of the environment that we find them in, we have to grow them on cultured media. Well, the media that we have, that nutrient-rich media that we want to grow them on, has to mimic the environment which they're in. So if we're looking at saltwater microorganisms, we need to grow them on media that contains salt. If it's freshwater, we wouldn't want to have high salt because that could kill the microorganisms. So we have this enriched culture in order to grow specific microorganisms. Or if we're taking microorganisms from the soil where we have a really high biomass concentration of microorganisms, so many microorganisms, and we want to reduce how many are in there, we can use something called selective growth media, which we'll talk about later, which can reduce some of the microorganisms within the media. Okay. I want to talk a little bit about some of the organelles that we find in cells and how they've developed. So eukaryotic cells themselves have membrane-bound organelles. Some of those are chloroplasts and mitochondria. Here we see the plant cell has chloroplasts. That's where photosynthesis takes place. And they also have in red mitochondria. Here we see mitochondria in animal cells, fungi, and protists as well. So mitochondria is in plants, animals, fungus, and protists. Chloroplasts themselves, we only find in plants, and that's actually what makes them autotrophic, meaning that they're actually making their own food, where we're not autotrophic. We have to gain our food from somewhere else. We're heterotrophic. Now, when we look at these membrane-bound organelles, first of all, we can see the size of them. The size of them is quite a bit smaller than their eukaryotic counterparts. And actually, the size of them is more similar to prokaryotic organisms like archaea and bacteria. Size-wise, very similar. Another interesting fact about the chloroplast and mitochondria is that they actually have two membranes. They have a double membrane system. So what can happen when cells, unicellular organisms, are consuming other cells? They engulf them in a process called phagocytosis. So it takes in the other organism. When it takes it in, it wraps its membrane around it and pinches off. And this is how we could see that we could have two membranes on top of one cell. So the endosymbiotic theory, which is explained in this image here, is showing us that actually chloroplasts and plant cells came from cyanobacteria, a photosynthetic microorganism that was engulfed and taken into the cell. Normally, when things are engulfed and taken into the cell, they are consumed, broken up, and the energy is used from them. But in this situation, we see that the chloroplast was beneficial for the plant cell. This chloroplast was all of a sudden feeding the cell, giving it glucose, giving it the food that it needs. So algae can have this as well. Beyond that, the microorganism, the cyanobacteria, benefited from being inside the cell because now it was no longer subject to predation from other organisms. It's no longer going to be consumed by other organisms. So it has a safe home at this point. It is completely protected from the outside world. So the chloroplast actually even has its own DNA, and its DNA is similar looking to what we would find in prokaryotic organisms. So after many different relationships that we see with these cyanobacteria, proteobacteria, as the primordial existence of these mitochondria and chloroplasts, we assume that they were picked up by the cell in a process called endosymbiosis. So endo being on inside, and symbiosis meaning it was a mutually beneficial relationship. For mitochondria, on the other hand, we may know these as the powerhouse of the cell. They're the ones making energy for the cell. So when they were consumed and not broken down, we saw them as basically a powerhouse for the cell, making additional energy for the cell. So the cell benefited from the energy, and the mitochondria benefited from having a home where, again, it was no longer subject to predation from other organisms. Now, back when I was in school over 20 years ago, this idea of endosymbiosis was a side note in our books. Today, I love it because we have large parts of our textbooks dedicated to the endosymbiotic theory because we've seen endosymbiosis live in action. They've been able to see other microorganisms consume, engulf, other microorganisms, smaller ones, so eukaryotic cells, taking in smaller prokaryotic cells, and instead of breaking them down in the traditional pathway, actually allowing them to live and forming a symbiotic relationship. So what this means for us is that all of our cells, as we have animal cells, have mitochondria within them. And these mitochondria were primordial proteobacteria, respiring bacteria, bacteria that could produce energy for the cell. So in all of our cells in our body, we can have upwards of 100 mitochondria. We have essentially these old bacterial cells, and they can divide naturally within our cells, and they have their own DNA as well. So if we look at if we're outnumbered by our own cells, we are definitely outnumbered by mitochondria in cell number inside of our own cells. Okay. Let's go ahead and look at microorganisms and what we're learning to understand about them. So we look at the three domains of life. We're going to look at eukaryotic, prokaryotic, and archaea. Archaea actually were thought to be they were thought to be bacterial in nature. So we have archaea, bacteria, and eukarya. because they were thought to be bacterial cells. They were just overly generalized and classified with bacteria for a long time until Carl Lewis came along and actually created a new category for archaea that is outside of what we see of just bacterial cells. When we look at the tree of life, we're going to see bacteria and then archaea spitting off and eukarya spitting off in another direction. Archaea actually have similarities to eukaryotic cells and prokaryotic cells. Let's go ahead and look at the DNA of organisms. DNA is extremely important because that's how we classify them down to their species level. Their DNA is their blueprint. Looking at the discovery of DNA, we have Rosalind Franklin in the 1950s who was doing crystallography imaging. So this is actually looking down the structure of a DNA double helix, and we can see down this helix and we can see the bonds between them. So this would be the base pair bonding on the inside, and the outside we would have that phosphate backbone. Watson and Crick later were not only able to take Rosalind Franklin's data, but other data, compile it all together, and build the first structure of the double helix that we see here, which was pretty chemically accurate, the way that they built together the bonding pattern of the DNA double helix. And while this picture seems quite old, what we see over here, this is actually Dr. Watson. I was able to meet him maybe 15 years ago or so. So he was still doing science as well at the time. Okay, looking at DNA, we're going to be talking a lot about bacterial DNA and how it builds the structure of the cell of bacterial cells. And we're going to see how it's a little bit simpler than our DNA in our body. Okay. Looking at the structure of DNA, molecular techniques have emerged for sequencing. So we've sequenced the entire human genome. This project started to understand the human genome in the 90s, and it took a very, very long time to sequence the human genome. The only reason why it got finished is because of advances in molecular techniques, where this is something we can do today. We know we can do something like 23andMe and send out our DNA. And have it analyzed very quickly. And this is because engineers and biologists were able to work together to develop instrumentation that has really pushed the frontier of molecular biology forward. So we have Sanger sequencing that we'll talk about later as well as a way to understand the code of life. So here we see the code of life. It's made out of 4 bases, C, T, A, and G, if we're looking at DNA. And we're able to look at this and read it almost like a book. We can know the sequence of these letters and what genes they make. Another good example of sequencing was that in 1995, they were able to sequence the genome of Haemophilus influenza. So it was the first one as a microorganism. Haemophilus influenza had a very, very small genome, so it was easy to do versus something like a human. Okay, at the end of these lecture notes, you're going to come across these questions. So I'll read the questions and then you can go ahead and hit pause to answer the question, and the answers will be on the following slide. So these questions are a great comprehension check. So the first question is, you discover a microbe that lacks a nuclear membrane. You found the microbe growing in a boiling sulfur spring in Yellowstone. Based on the cell structure and extreme environment, you hypothesize this microbe is likely, and you have the answers here to look through. So you could pause now. And we see it's going to be archaea. So some of the hints about it being archaea would be the fact that this lacks a nuclear membrane. So archaea and bacteria lack nuclear membranes. A virus would not be considered a living organism. So here we see the microbe is growing, so we can exclude virus. Fungus would have a nuclear membrane, and protozoa would have a nuclear membrane. So the other thing that divides bacterial cells and archaea is the fact that we found it in extreme environment. So archaea are found in extreme environments. Bacteria can be as well, but most likely archaea. Okay, question 2, which of the following is characteristic of a virus? And again, this is where you would pause. Okay, The answer to this question is that viruses are unable to reproduce independently. So again, viruses are dependent on other cells for reproduction purposes. They're using their molecular machinery, like a DNA polymerase, to make new DNA. Question 3, assume that the theory of spontaneous generation is true. Predict the results if you were to repeat Pasteur's experiment shown below. So what you would find if spontaneous generation is true, which it isn't, but if it was, you would find that microbes would grow in the flask. They would poof into existence into the flask here. Question 4. You're attempting to prove that bacterium A causes a respiratory disease in laboratory mice. You have isolated the bacterium from sick mice and grown the bacterium in a pure culture. What is the next step you would take to fulfill Koch's postulates? We'll go ahead and move on to the answer. It says that we would inoculate a healthy laboratory mice with the pure culture. So once that pure culture is obtained, again, you would take that, inoculate a healthy mouse, see if they succumb to the same signs and symptoms. Question 5. Which type of relationship between cells leads to the evolution of eukaryotes and multicellular life? So this would be endosymbiosis. That's the theory of how the chloroplast and how the mitochondria came about. And that's what led to eukaryotes being different from prokaryotes because they have membrane-bound organelles like the chloroplast, like the mitochondria. And this later led to support multicellular life. Okay, question 6. Which of the following statements about bacteria are archaea true? Bacteria and archaea are true. Okay. It says the cells of both bacteria and archaea are prokaryotic in design. So that's correct that when we look at bacteria and archaea, they both have simple architecture of being a prokaryotic cell. They don't have any membrane-bound organelles, and they're very small. So they're very similar, and that's actually why originally they were classified both in both as bacteria, and then archaea branched off later when we started finding differences between them. What we find at the end of the lectures here is a section objectives. This is what you can use as a study guide, and this is recommended to do after listening to one of the videos here. So from germ to genome. So what you can see here, you can read through section objectives, and I would fill these out. So this is your study guide, and this is meant to be done after the lecture. So you can go through these section objectives and fill them out to make sure that you understand the lectures. And that concludes the first lecture.